topic is plant growth and development. Please reply. Yes, good evening, Shivam. Reply in the chat that if voice is clear or not, so that we can start the topic. Good evening, everyone. Reply in chat box if my voice is clear. Hello, good evening. Hope you can hear me properly. Okay, we are moving ahead. So our today's topic is plant growth and development from the unit that is plant physiology. Okay, last topic of the plant physiology. So before jumping in the topic, I want to insist all of you that follow us in any of this link. You can follow us on Facebook, YouTube, Instagram, Telegram, anything you want. Okay, you can follow us on any of this link. You can even download the Spark Academy app from the Play Store and then enroll any of the courses, and you can have, after joining the course, you can have free access to study material, free previous year question papers, question bank, free videos, exclusive live class are continuously going on, regular live class, revision class of okay. So it is continuously going on. So you can join that live class and you can ask your doubt in that live class, during that live class. Even after that, you can check out the video in your app. Okay, so we are taking chapter wise test also, full length tests are also we are taking. If you have any doubt in any of the topic, you can tell in the chat bot. You can write in the chat box. We can have a doubt clarification sessions afterwards. Okay, we can arrange doubt clarification sessions afterwards. So I'll insist again all of you to download this app from the Play Store, Spark Academy app, and then join any of the courses. Okay, this is how live classes are going on continuously. You can join any of the live classes. And we are continuously giving you assigning assignments to you on daily basis. We are assigning assignments. Okay, in that assignments, in those questions, if you have doubt, you can ask for that also. We can have a doubt clarification sessions. So this is how the right now it is on 50% discount, so you can join any of the course using the Spark 50 code. So you will get 50% discount in any of the courses. Now let us begin with the topic, that is plant growth. How plant growth is plant is starting its life. Okay, see, it is from zygote. First a zygote will be formed, then from zygote it will divided into 2 cell, then 8 cell embryo, then globular embryo. This is how a full-fledged plant is grown. So zygote develop into a mature plant through growth and differentiation, forming the roots, leaves, branches, flowers, fruits, seeds, and eventually the plant die. Okay, so plant growth is generally indeterminate. The plant growth continues throughout the life due to presence of meristem in the plant. Okay, so continuously growth is occurring in a plant body. So meristematic cells have a capacity to divide. They have capacity to divide and they have capacity to continuous divide and the growth, where new cells are always added to plant body by activity of meristem, is called open form of growth. See, you might have we might have discussed in the anatomy part and in the of this flowering part. Okay, see, in that there are two types of growth. Okay, if vascular bundle is present, so between vascular bundle, if it is divided with this cambium part, cambial ring, then we can say this cambium ring will again start growing. It will differentiated. It will start growing meristem. It is a type of meristem. So in that case, there will be secondary growth. So secondary growth, plant is open for secondary growth. Okay, plant is open for the secondary growth. So that means it open form of growth is there. Okay, if this cambium ring is not present between vascular bundle, between xylem and phloem, and then the growth, secondary growth is not there in that type of plant. Okay, so plant growth generally is indeterminate. Okay, see how it is. Vascular cambium, between that vascular cambium is present, this vascular cambium start growing. It will divide in both the side. Okay, it will send continuously cut off the cells, new dividing cells in both the side of the root as well as shoot. Okay, so this is how this is primary growth. So it occurs due to the root apical meristem and shoot apical meristem. It causes the elongation of plant along the axis. Secondary growth in case of plant, it is in gymnosperms as well as in case of dicotyledon plants. So it occurs due to lateral meristem, vascular cambium, cork cambium, and this causes the increase in the girth of the organs. Okay, thank you, Kulsum. Good evening, all of you. Next, plant growth is measurable. So at cellular level, we can measure the growth of plants. So at cellular level, growth occurs due to the increase in the amount of protoplasm. For example, this is a single cell. At single cell level, only protoplasm is increased. So at cellular level, it is growth, that means increase in protoplasm. Increase in protoplasm is difficult to measure. So we can't measure this type of increase in the protoplasm. So growth is measured by parameters like increase in the fresh weight, dry weight, length, area, volume, cell number, etc. Cell number, a maize root apical meristem can produce more than 17,500 new cells per hour. So we can measure the cell number. Okay, cell size. Cell is in a watermelon may increase in the size by this times. Okay, so length, growth of the pollen tube. We can measure the growth of the pollen tube. We can measure the length of the pollen tube. Okay, so we can say that if it is growing, that means length is increasing. Surface area, growth in a dorsiventral leaf. See here, this is a dorsiventral leaf of dicotyledon plants. So next, plant growth, phase of plant growth. So meristematic phase, first phase of plant growth, that is meristematic phase. It occurs in the meristem, at root apex and shoot apex. So mostly at the apical region. Okay, the cell is in this region have rich protoplasm and large nuclei. Okay, it will be having large nuclei. Cell wall are primary thin. Whoever is continuously growing, if you have asked, cells which are continuously growing, they have large nuclei as compared to other cells, and they have thin cell wall. Okay, and cellulosic with abundant plasmodesmata. So it is made up of cellulose. If see, we might in that tissues, type of tissues and type of cells in that tissues, we have studied that in that case, if there is lignified or cells are secondary. What is the secondary cell wall? That means that is not made up of, outer side it is primary cell wall which is cellulosic, and inside there is deposition of lignin. So in that case, that cell becomes dead. But continuously growing cell is thin and having large nuclei, and the cell wall is made up of cellulose. Okay. Next phase is elongation. So after continuous growth, see, look at here. First it will start growing, then it is elongation. So it occurs in cell proximal, next away from the tip to the meristematic zone. Cell have increased vacuolation size and new cell wall deposition. Maturation phase. Third is maturation phase. It occurs in the cell further away from the apex. The cells attain maximal size. Now cell is at maximal size. Here they are continuously growing. That means continuously dividing cell. The size will be smaller. Again this became longer, and here it reaches maximum size. Okay. The term of wall thickening and protoplasmic modification. Okay. Size in terms of the wall thickening. If it is divided into any secondary tissues, okay, permanent tissue, if it is differentiated into forming permanent tissue, then it may get thickening at the cell wall and it becomes dead after that. Okay. So next is growth rate. Here R is relative growth rate. R is relative growth rate. It is also is the measure of the ability of plant to produce new plant material. Efficiency index. It is known as efficiency index. Hence, final size W1 depends on the initial size W0. So initial size it is W0. Final size it is W1. Quantitative comparison between the growth can be made in two ways: absolute growth rate, measurement and comparison of total growth per unit time, and relative growth rate, measurement of the growth of a given system per unit time expressed on the common basis like per unit initial parameter. So here both leaf A and B have increased their area by 5 centimeters square in given time to produce A1 and B1. Okay, see here it is increasing like this. Okay, in every area. Here it was smaller and growth is more. Okay, we can say here or it also increases in size. If we measure directly size, we need to measure the initial size also. Okay, so this final size depends on initial size. So growth depends on the initial as well as final. Hope you are clear with this. So next is plant growth. So conditions for growth. Essential elements for growth are, so water. Conditions for growth includes water, oxygen, nutrients, temperature, light, gravity. Okay, so we'll discuss one by one. Again, I am repeating: water, oxygen, nutrient, temperature, light and gravity. So first is water. For cell enlargement, turgidity of cell helps in the extension of the growth. Water provides medium for the enzymatic activity needed for growth. So water, you all know that water is universal solvent. Okay. So all enzymatic activity, all soluble nutrients, getting nutrition is provided by water. So for cell enlargement, turgidity, see, even in case of this stomatal pore opening and closing, okay, this pore opening and closing is also because of this guard cell and turgidity of this guard cell. Turgidity of guard cells. So how they become turgid? Just with the help of water. If they absorb more water, then they become turgid, and because of turgidity of this guard cell pore, stomatal pore will open. When there is water loss from the guard cell, when they loses water, it becomes flaccid, and because of the flaccidity of the guard cell, stomatal pore closes. It will be like this. Okay, now they are flaccid. Now they are flaccid, and the spore is closed now. So this is how gaseous exchange. They can control gaseous exchange. They can control water intake and so on. So next is oxygen. It helps to release metabolic energy for growth. Oxygen is required. Oxygen requirement of plant is provided through photosynthesis only. One of the product of photosynthesis is oxygen. Okay, so plant will get this oxygen from their cells only. So next is nutrient, micro, whether it is macronutrient or micronutrient. In this unit only, nutritional requirement we might have you might have studied. Okay. So nutrients that may be macronutrient or micronutrient, that means requirement is large, and here they are required in small quantities. So depending on the quantities, there are two types of nutrients, there are two types of elements. If they require in large quantity, then they are known as macronutrient. If they are required in small quantities, then micronutrients are needed for the synthesis of protoplasm and act as a source of energy as well. Next is temperature. Plant have an optimum temperature at which growth is maximum. Okay, you all know about the word optimum. Okay, we can tolerate, see, plants can tolerate some of the fluctuation in temperature, but at some point of the temperature, their growth is maximum. That some point where the temperature where their growth is maximum, that temperature is known as optimum temperature, optimum growth conditions for plant. Water should be optimum, oxygen should be optimum, nutrient should be optimum, temperature should be optimum for the maximum growth of the plant. So deviation from this range could be detrimental to its survival. Okay, next is light and gravity. So affects certain phases and stages of light, also affects intensity of light, affects certain stages and phases of the growth. Like light intensity affect the photosynthesis part also. Next is differentiation, de-differentiation and redifferentiation. See, we need to understand this word. Differentiation, that means from meristem to permanent tissue. Okay, then after becoming permanent, some of them again start differentiation, again become into converted into meristem. That is known as de-differentiation. Okay, from meristem, it will differentiated into permanent. Okay, after some point of time, after some period of time, again this will become, it will converted into meristem. Then this is known as de-differentiation. Okay, then again it gets differentiated to become permanent. Then it is known as redifferentiation. This is how these three processes are going on. Okay, we'll start with the differentiation part. Differentiation is the process in which the meristem cells, root apical and shoot apical, and the cambium differentiate and mature to perform specific function. Okay, here they become mature. So from babies to maturity. So matured person. So that means differentiation. And this cell wall protoplasm undergoes structural changes and capacity of cell division is lost. Okay, there is no more cell division occurs after it is differentiated and after it is becoming mature. Okay, for example, loss of protoplasm to form a trichory element. They also develop very strong elastic and lignocellulose secondary cell wall. See, I have explained you. See, in this meristem cell, if this is primary, the cell wall is very thin. Cell wall is thin here. Okay, when there is deposition, when it is differentiated to make any permanent tissue, so there will be deposition of lignin Either it is lignin or it is pectin. Okay, in case of pectin deposition, it will be not completely dead cell. It will be not forming completely dead cell. Okay, so here lignocellulose, that is deposition, that will that is known as secondary cell wall, to carry water to long distance even under the extreme tension. See, this lignin deposition is needed to make it very strong, elastic, and elastic. Okay, and because they need to travel, it need to carry water to the long distance even under the extreme tension. Okay, so next is, under certain condition, living differentiated cell regains the capacity to divide. Okay, so that means divided cell. Okay, newly arrived cell, divided cell, that is meristem. Then it is becoming mature, so it is losing capacity. Now it is not having any capacity to grow, to divide. Okay, after some period of time, if it is regaining this capacity, okay, regain of this capacity to divide. Okay, regain the capacity to divide, then this is known as de-differentiation. Okay, this is called de-differentiation. So formation of meristem from we have studied this interfascicular cambium, cork cambium from the differentiated parenchyma cell. Okay, so this parenchyma cell, yes, ready? Do you have any doubt? So de-differentiation, that means again they are regaining capacity of division. Okay, so that means formation of meristem meristem again in form of interfascicular cambium and cork cambium from differentiated parenchyma cell. See, this cambial ring is formed. This is interfascicular cambium and fascicular cambium. Okay, so finally there will be formation of this ring. So again they will start dividing, and the cells cut off in this side, in both the sides. Most of the cell cut off in this side towards the pith. Okay, so there will be secondary growth. There will be formation of secondary xylem and secondary phloem in that case. Okay, and bark formation also will be there because of the cortex region, because of the cork cambium. Okay, so this is how secondary growth, it is result of the de-differentiation of permanent tissue. Okay, next is redifferentiation. So here de-differentiated cell can produce cells that again lose the capacity to divide, okay, but mature to perform specific function. This is called redifferentiation. So plant growth is open for this. So plant growth is open. It can be intermediate or determinant. Differentiation is also open because cells and tissue arising out of the same meristem have different structure at the maturity. Final structure at maturity of cell and tissue is also determined by the location of the cell. Cell position away from the root apical meristem, differentiated at root cap cells, and while those pushed up to the periphery mature as epidermis. Okay, this is how differentiation, de-differentiation and redifferentiation going on. See, differentiation is going on here. Okay, so de-differentiation, that means stem cell like state. It can redifferentiate again into formation of any type of cells. Okay, so redifferentiation, that is transdifferentiation, cell death and cell cycle. Okay, so next is plant development. Okay, now we have discussed about the growth part. Now we are moving towards the plant development. It is process that includes all changes in the life cycle of the organism from seed germination to senescence. That means it is dying. So it is falling. So it is the sum of the growth and differentiation. It is represented as follow. Meristematic cell, then plasmatic growth, expansion, elongation, differentiation, and maturation. Finally, we are getting mature cell. Okay, either it is going to the death, okay, deathbed, finally senescence and death, okay, or from here it will again becoming meristematic. Okay, from here it will start redifferentiation, dedifferentiation and redifferentiation. So this is how it is going on. So it is the process that includes all changes in the life cycle of an organism from seed germination to the senescence, sum of the growth and differentiation. So from zygote to 16 cell embryo to heart stage embryo, okay, then to juvenile stage, adult stage and reproductive stage. Finally, there will be formation of reproductive organs and all. So plant development. Plant follows different pathways in response to environment. In response to the environment or the phases of the life to perform different kinds of structure. This ability is called plasticity. Plasticity of plant, that means a different pathway. There is no particular pathway. It follows, different plant follows different pathways in response to the environment or phases of the life. Next is that is heterophyly due to the phase of the life. In cotton, coriander, larkspur, the leaves of the juvenile plant and mature plant are different in shape. See, this is juvenile plant and this is adult plant. You can see the difference between these two in their shape. Okay, so heterophyly due to the environment. This is example of buttercup and larkspur. So difference in the, if you grow and see, it may be difference in their juvenile stage and their mature stage. So because on the basis of their phases of their life, the shape is different. And on the basis of environment condition, if you grow this type of plant of buttercup in water and air, okay, if you grow it in water, there will be some different shape of the plant, of the leaves, and in air it will be of different shape. So it shows some different shape, that is known as heterophyly due to environment, heterophyly due to phase of life. Okay, so now plant development. Factors controlling the plant development. It may be intrinsic factors, it may be extrinsic factors. So intracellular factor, intrinsic, that means intracellular genetic factor, plant growth regulators, and extrinsic include light, temperature, water, oxygen, and nutrition. So plant growth regulator, first is intrinsic factor, in that we can consider plant growth regulators, that means hormones. Growth regulator, that means hormones. Wait a second, let me check the chat. Okay, so plant growth regulator, that means hormones. So plant growth regulators, those are known as PGRs, are small, simple molecules that regulate plant growth. Based on the function, PGRs are of two types. Plant growth regulators are of two types: plant growth promoters and inhibitors. Okay, they can promote the growth, they can inhibit the growth. Based on their function, they are of two types. For growth promoting activities like cell division, enlargement, entropic growth, and pattern formation, flowering, fruiting, seeds formation, there are three plant growth hormones which are involved in this as promoters: auxin, gibberellins, and cytokines. and inhibitors for inhibiting the activity like dormancy and abscission, respond to the wound and stress of the biotic or abiotic origin, abscisic acid and ethylene. Okay, so there are 5 hormones we are going to discuss today: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. Ethylene fits either of the group, but it is largely as growth inhibitor. Okay, sometimes it is fits as promoter as well, but mostly it is considered in inhibitor part. Next is growth regulator. First growth regulator, that is auxin. So Charles Darwin and his son Francis Darwin observed that the coleoptile of the canary grass responded to the unilateral illumination by growing towards the light source. So that is known as phototropism. If it is growing towards the light, that is known as phototropism. It was concluded that tip of the coleoptile causes the bending of the entire coleoptile. Okay. This Francis Darwin went isolated this FW went then isolated auxin from the tips of the coleoptile and of the coleoptile of oat seedlings. Okay, from the oat seedlings he isolated auxin. Okay, from that seedling he isolated auxin from the coleoptile of oat seedlings. Auxin were first isolated from human urine. Auxins are produced by growing apices of the stem and root from where they migrate to the region of their action. Okay, it is first produced here at the apical region, then it is migrate towards the where it is needed, where is the action. So production site is different and action site is different. You need to remember this point. Okay, you need to remember this point that auxin, in case of auxin, it is produced somewhere else and it is working, its working site, its action site is somewhere else. Okay, it migrates to the region where their action is required. Okay, next type of auxin, nature of auxin. Chemically, it is indole 3-acetic acid. So it is IAA, which we call as auxin. Chemically, it is auxin, indole 3-acetic acid, indole butyric acid. So there are 2 auxins, IAA and IBA. They are isolated from plants. Synthetic auxin is also there, that is naphthalene acetic acid and 2,4-D. Okay. So these are natural. This is also important to remember. IAA and IBA are natural auxin and NAA and 2,4-D are synthetic auxin. So if we want to give this hormone, if hormone supplementation to the plant, we can give in this form. Okay, these are NAA and 2,4-D. We can give in form of fertilizer to the plant. So these are synthetic auxins. Plant growth regulators, in that first auxin, we are discussing that only, so it is continuing. Function of auxin is to initiate rooting in the stem. Initiate rooting in the stem cutting for the plant propagation. Okay, when the stem is cut and from that rooting is initiated. Okay, so this is from the stem cutting for the plant propagation. When you are doing propagation of any plant, you are cutting that stem. In case of rose, we are doing that. Okay, you cut off the apical region. Okay, you cut that stem and you are again propagating it. In that case, again rooting of that propagated plant will start because of the auxin. Okay, it promotes flowering. An example is pineapple, to prevent fruit and leaf drop at early stage. Okay, it is young and growing leaf, it will not fall off because of this auxin, action of auxin. Okay, young leaves, young fruit will not drop because of action of auxin. Promote the abscission of the older leaf and fruits. Older leaves will fall off. Induce parthenocarpy in tomatoes. They are used as herbicides. 2,4-D is used to kill dicot weeds. It does not affect monocot plants. It is used to prepare weed-free lawns. Okay, when you are growing a crop, or when you are taking crop of corn, it is monocot. Okay, you don't want any dicot weeds to grow beside it. Okay, you just want to promote your monocot to grow. In that case, if you are inserting or spreading their 2,4-D. So this 2,4-D is synthetic auxin. It is acting as herbicide for the dicot. So dicot weed will be killed in that case, and you will have prepare weed-free lawns. Then last is control xylem differentiation and help in the cell division. Okay. Next, plant growth regulators auxin continues. In higher plant, the growing of apical bud inhibits the growth of lateral buds. Okay, it is known as apical dominance. Here, it is known as apical dominance. In higher plant, growth of apical bud is inhibited. Okay, apical bud here, look, you can see here, this apical bud will not grow. It will remain as such, and because of that, lateral growth of plant will be there. Okay, so this is known as apical dominance. Here apical buds will not grow. Lateral buds will start growing, start branching. This is known as apical dominance. I am again repeating it. In higher plants, growing apical buds inhibit. See, this is growing apical bud. Okay, after formation of bud here, there will be auxin secretion there. Okay, so because of that auxin, it will be in the phenomenon known as apical dominance. So it will not grow. Lateral axillary buds will start grow. So removal of shoot tip result in the growth of lateral buds. It is apical applied in the tea plantation and hedge making. Okay, so in that case, this apical tip is removed. So otherwise, it will grow, continue on growing. Okay, if you remove this tip, then there will be a starting of secondary lateral growth. Okay. So next is gibberellin. These are acidic PGRs. Gibberellins are acidic plant growth regulators. Kurosawa treated the sterile filtrate of Gibberella fujinkuro, a fungus that causes Bacani, a disease of a foolish seedling in the rice, of which fungus to healthy rice seedling. As a result, it showed the symptoms of Bacani disease. Later, the active substance were identified as gibberellin. There are more than 100 gibberellins. It is named as GA1, GA2, GA3, etc. in fungi and higher plants. Gibberellic acid is one of the first plant growth hormones. Okay, next, functions of gibberellic acid or function of gibberellins. They causes increase in the length of axis. So they are used to increase the length of the grape stalks, to elongate improve the shape of the fruit such as apple. They delay the senescence, so the fruit can left on the tree to extend the market period. Okay, if we need to, otherwise plant will fall. Auxin production will start. If they are all older, it will fall off from the plant. So in that case, if you increase gibberellins, that will delay the senescence. So fruit can left on the tree to the extent of the market period. Some of apples you have sold, some are still remaining on the plant. So it will be fresh. So you can say it is farm fresh apples, farm fresh fruits. So GA3 is used to speed up the malting process in sugar. Next is sugar can store sugar in the stem. spraying sugarcane crop with the gibberellin. Spraying sugarcane. Wait a second. Spraying sugarcane crop with the gibberellins increase the length of the stem. Okay, you will have longer sugarcane in that case. It increases the yield by as much as 20 tons per acre. So spraying juvenile conifers with the gibberellic acids hasten the maturity period. It leads to early seed production. Okay. In that case, if they are young and they are not reaching at the reproductive stage, in that case, if you spray gibberellin over that, juvenile hasten the maturity period, and they will start formation of seeds. For blotting, internode elongation just a period to flowering in beet, cabbage, and making plants with rusty habit. Next is cytokines. Skog and co-workers observed that for internodal segment to the tobacco stem, the callus, okay, callus, that means undifferentiated cell. It is not differentiated, it's a mass of the cell which is, that means you can say in other words, mass of stem cells type of cell. In plants, they are not stem cell, but we can say stem cells type of cell. So mass of this stem cell type of cell, they are known as callus. Okay. So in a petri dish, if you start growing a plant cell, you will have this type of mass of cells growing everywhere. This is your callus. In that case, if you give growth hormones, it will, and if you plant some of few of cells in soil, it will start growing. It will start growing in presence of PGRs. Okay. So in this case, the Skog and co-workers observed that internodal segment of tobacco stem, the callus proliferated only if the nutrient medium was supplemented with extract of vascular tissue, vascular tissues, yeast extract, coconut milk, and DNA. Okay, here this is your nutrition supplement. Here it is coconut water. In that, if you are adding DNA, vascular tissue, and yeast extract, only in this medium, if you want to grow your callus. Okay, if you want to grow your callus to the tobacco plant, to the segment of the tobacco plant. So Skog and Miller later identified the crystallized active substance termed as kinetin. So cytokines were discovered as kinetin, N6-furfuryl laminopurine, an adenine derivative from the autoclaved herring sperm DNA. Kinetin does not occur naturally in plant. Kinetin does not occur naturally in plant. This is kinetin which is not occurring naturally. Okay. Cytokines. Zeatin from the corn kernels and the coconut milk is the natural substance which is cytokine-like activity, which has cytokines-like activity. There are some synthetic compounds which cell division promoting activity. Natural cytokines are synthesized in region of rapid cell division. So in rapid cell division at the apical region, at the bud region, okay, or at the meristematic tissue. So cytokines are synthesized at the meristematic tissue. The next function of cytokine they play role in cytokinesis. Okay, cytokinesis, you remember during the cell division. Okay, they help to produce new leaves, chloroplast in the leaves, lateral shoot growth, adventitious shoot formation. They help overcome the apical dominance. Okay, they help in overcoming the apical dominance. If you don't want apical dominance, in that case, you need to remove that apical bud. Okay, so then you will get lateral branches. But if you want to overcome without cutting it, then you can help with the cytokines. Okay, so this is how lateral growth will occur. So they promote nutrition mobilization with help in the delay of the leaf senescence. Next, ethylene. What we are seeing right now, mangoes, carbide free mangoes, okay, or carbide wali mangoes. These mangoes are because of this carbide. What is this carbide and what is this carbon? That is ethylene. Look at this chemical formula of ethylene. Carbon is there, so it is, we call it as carbon. So cousins confirmed the release of volatile substance from the ripe oranges that hasten the ripening of stored banana. Later, this substance was identified as ethylene. Ethylene is simple gaseous, so it is in gaseous form. Remember, gaseous PGR, that is ethylene. It is synthesized in large amount by tissues undergoing senescence and ripening fruit. Okay, if you don't pluck those fruits, they will fall off from the tree because of ethylene. Okay, if they are ripe, plant will not. So they are older leaves. They are older. They are older leaves. They are older. What we call fruits. So ripened fruits. So plant will not carry this fruits and it will fall off. It will result in the senescence because of production of ethylene. So in that case, you need to spray gibberellin. So plant will hold this for some more time for to increase your market time. Okay. So ethylene function, it influences the horizontal growth of the seedling. Okay, horizontal growth of seedling, swelling of the axis, apical hook formation in the dicot seedling. It promotes the senescence and abscission of the plant organs, especially of leaf and flowers. It promotes the fruit ripening. It enhances the respiration rate during the fruit ripening. So this is called respiratory climatic. It breaks seeds and bud dormancy, initiates the germination in the peanut seed. Sprouting of potato tubers also because of ethylene. Some more functions: promotes the rapid internode and petiole elongation. Rapid internode and petiole elongation in the deep water rice plant. Okay, it helps leaves and upper part of the shoot to remain above the water. Promotes root growth and root hair formation. This increased absorption surface used to initiate flowering for the synchronizing fruit set in the pineapple. It also induces flowering in mango. Okay, so it is widely used in agriculture. The most widely used source of ethylene is ethepone. Is an aqueous solution. See, mostly it is in gaseous form if it is natural and if it is naturally secreted by plant. But this ethepone is it is aqueous solution, readily absorbed and transported within the plant and release ethylene slowly. Okay, when you spray it, it will readily absorbed and transported within the plant and released. Then finally there is slow release of ethylene will start. So ethepone hastens the fruit ripening in the tomatoes and apples and accelerate the abscission in flowers and fruits. It promotes female flower in the cucumbers and thereby increasing the yield. Okay, if we spray this, there will be this unripe tomatoes will start ripening because of slow secretion of ethylene by plant after spraying of this ethepone. Okay. If you spray it over the cucumber plant, then there will be yield it promotes the female flowers, then there will be finally more yield of cucumbers. Next, last growth hormone, that is growth regulator, that is abscisic acid. During mid-60s, it was reported three kind of inhibitors, inhibitor B, abscissin 2 and dormine. They were chemically identical and known as abscisic acid, 3-ABA. Inhibitor B, abscessin 2, and dormant. ABA is a derivative of carotenoids, which gives orangish color to the plant leaf and plant product. It regulates abscission and dormancy. Functions: it acts as an inhibitor of the plant and metabolism. It inhibits the seed germination. It stimulates closure of the stomata in epidermis. It increases the tolerance of the plant and various kind of stresses. It has important role in the seed development. Okay. So interaction of PGRs. PGRs play individualistic and synergistic role. Such role is complementary to antagonistic. Factors influencing PGRs are intrinsic and extrinsic. Okay, extrinsics includes light and temperature. Photoperiodism is there for light. So long day plants, short day plants, day natural plants are included in this type. Okay, role of light and temperature on flowering in that photoperiodism. Okay, you can see this type of things because of the photoperiodism, because of the sunlight. Okay, then vernalization. Then vernalization, again I will remind you all of you to join us, download the app and join any of the crash courses. Okay, now quickly we will move towards the question answer part. You all are ready. I hope you all are ready for the answering questions. So first question is, in plant growth is restricted to certain regions or structure. It is irreversible. It is change. What is growth is, that means change in size, or all of above are correct for the growth. If we want to describe growth in plants, which of these is correct? Answer: restricted to certain region or structure, irreversible, change in size, all of above. Come on, faster. Kulsum, Hafsa, all of you. I'm waiting for your answer. You should write with your question number 1A, 1B, 1C, 1D. I'm waiting for your answer. In plants growth, that means what? It is restricted to certain regions or structure. Yes, it is restricted. I will give you answer because we are having only few minutes left. So we need to solve all the question answer in that. Okay. Shruti, Sazia, it's not correct. Think again. Restricted to certain region, irreversible, change in size. Yes, change in size is also true for the plant growth. Okay, if it is cellular level, then change in size. Yes, it is irreversible. Okay, it is restricted to certain region and this or structure only. I mean to the meristem only. So all of above is the correct answer. Okay, next question. Growth is primarily affected by two climatic factors: light and temperature, temperature and relative humidity, light and wind, rainfall and temperature. Answer for the second question: 2A, 2B, 2C, 2D. What is your answer? Quickly. Sazia, Shruti, answer. Kulsum, all of you. Quickly. I want quick answer. Only 6 minutes left. Growth affected by which of these two climatic conditions? I want quick answers. Come on fast. Yes, correct. Correct, Sazia. It's 2A. Light and temperature. Next, come on fast. Which of the following instruments can be used to record plant growth by seconds? Instruments for measuring plant growth. Correct, Shruti. Now answer for the third question. I will give answer for this question. This is crescograph. Crescograph is the instrument which is used to measure, record the plant growth by seconds. Okay, now answer for the fourth question. Maximum growth rate occurs in which phase? Which growth phase? See, growth curve will be like this: lag, exponential, stationary, and then decline. That is senescent phase. So this phase, lag, this is log, that is exponential, and here it is stationary, and then senescence. Yes, 3D is correct answer. Rohini, all of you answer for the fourth one now. Which phase growth rate occurs maximum? Growth rate is maximum in which phase? Continuously dividing cell. Growth phase maximum. Wrong, wrong, wrong. Sazia, Shruti. It is in exponential phase. This is continuously growing phase. Continuously growth rate is maximum here. In that phase it is maximum, you are saying? Yes, Sazia, now you are correct. Exponential phase. Okay, next, fifth, apical dominance. Only 3 minutes left. Apical dominance means suppression of growth of apical bud by axillary buds. Suppression of growth of axillary buds by the presence of apical bud. Yes, this is correct answer. If apical bud is there, growth is in apical region will be more. In this case, you need to remove, cut off the apical bud. So that means apical bud is dominating here. So correct sentence for the apical dominance is suppression of growth of axillary buds with the presence of apical bud. Next is for sixth question, maximum amount of growth in root occurs. Maximum amount of growth in roots. Maximum in roots occurs at which stage? In presence of light, at its apex, behind its apex, in the presence of soil. Shruti, answer for the sixth question. Sixth question. Everyone, sixth question. Sixth question. No, not at apex, Sazia. In root, you have forgot here it is root cap. Okay, root cap is present. So, root cap is apex of that. Okay, here there will be continuous growing cells. Okay, this is meristem of the root. So, it is behind the apex. So, correct answer is 6C. All of you are wrong. So, 6C is correct answer. Next, which of following plant material widely used in preparation of culture medium? Widely used in preparation of culture medium. It is coconut milk which we are using, so it is Cocos nucifera. The stimulus for the geotropism in root is perceived by what? It is by root cap. Okay, geotropism in root, it is because of root cap. Next, the method that renders the seed coat permeable to water so that embryo expands in the is not physically retarded is called. What is this method called? It is called scarification. Growing points present at the at only growing points present at only meristematic cells. Next, the growth of the plant body takes place. Growth of the plant body takes place where? Okay, for 11th answer is both apical and lateral meristem. Okay, last, the pineapple, which under natural condition, which is difficult to blossom, has been made to produce fruits throughout the year by application of which? Okay, fruits are produced throughout the year by application of what? By application of NAA-N2,4-D. Okay, that means synthetic auxin. Okay, thank you very much all of you for joining this session. Okay, we'll meet again. Thank you very much all of you for joining this session.